And we're back. So this whole section is just going to focus on blood types and how they react with each other. So dealing with being able to get a blood transfusion and how that can interact between a mother and a fetus. So our blood types was determined by Carl Landensteiner. We focus mostly on the ABO blood types because those are our more reactive ones. There's actually a ton of different glycoprotein markers on our blood cells, but they don't have the reaction like our ABO, and then the positive versus negative for the RH factor. Those are the more reactive ones, and so those are the ones we focus on more because those are the ones that are going to cause us a problem. So our blood types are based on the interaction between our cell surface antigen and our plasma antibody. So they have two different vocabulary names for them, specifically dealing with the blood, and they're very close to each other. So we're going to have to pay attention to the difference. Okay, so our antigens, these are our cell surface markers on our red blood cells. So these are the ones that once detected in a donor's body can cause an immune response, as in this is not the right type of blood. And so that's how they're being able to distinguish, wait, this blood doesn't belong to us. It has a different glycocalyx. So it's actually going to generate an immune response because our body detects that this is foreign blood. This is why we have to get either the same blood type that we are, or for a few of our blood types are able to receive all the different blood types because they don't have antibodies that would find out, like, hey, hold on, without those antibodies, they wouldn't be able to find those antigens. So they're able to receive almost any type of blood because they don't have the antibodies that can recognize these antigens. Okay, so we get a foreign antigen, a foreign cell surface marker, generating an immune response. So the specific markers that are on a red blood cell that could elicit such a immune response, so generate one, is called a agglutinogen. So remembering the gen portion for our antigen. So these are cell surface markers specifically for a red blood cell. So this is what we use for our blood typing, because when those cell surface markers react to antibodies that we can place in the sample, then we know what type of blood type it is. So that's what they're doing. If you ever give blood, they're going to test for your blood type so they know which type it is, so they can mark it down so they know who to give it to. Okay, so on your, whatever this thing is, study guide, number 16, define agglutinogens. There's your definition. So these are the cell surface markers on the blood. This is the basis for your blood type. So it determines what your blood is because of that marker on the blood cell. Now, the antibodies are in your plasma. So whatever blood type you have, your blood creates antibodies against the other types. So that's how they're able to detect a foreign blood type that has gotten into the body. If you've given a transfusion and then maybe the bag was marked wrong, and so you're going to have an immune response. So these antibodies, again, they're produced by the same white blood cells that would, okay, not exactly the same, but same type of white blood cells that create your regular antibodies when you get a flu vaccine, when you just get sick. And so your body just has to make antibodies against that foreign pathogen. Okay, so these antibodies, they bind to cell surface antigens, whether it is foreign blood or a foreign bacteria or foreign virus. So marking that cell for destruction. So we have other types of blood cells. They are like Kill Bill Volume 7. Like their job is to just kill stuff. It's like, oh, foreign enemy, let's go get it. Okay, so when those antibodies bind, they form a antigen-antibody complex, and that marks it for destruction. So the antibodies specifically for our foreign red blood cells, they're all called agglutinins. So that is their specific name for the antibodies against the other blood cells. Remembering there is some blood types that actually don't make certain antibodies, and so they're able to receive more blood than other types. Okay, so still on that same one, define agglutinins. Ding dong, there we go. So these are the antibodies in the plasma against the other blood types that can form your transfusion reaction, so a mismatch if you get the wrong blood type. So these are going to do your immune response in that situation. So what is that immune response called? It's called agglutination. So this is... Okay, so this is where these antibodies, these agglutinins, bind to the antigens, also called the agglutinogens. When they bind together, they're going to cause a clumping of the red blood cells. So it makes everything thicker and sticky, and it keeps those red blood cells from circulating around. So that clumping is called agglutination. You'll see a picture of it later. Okay, so for our ABO blood type, the cell surface antigens, again called agglutinogens, we have one for antigen A, one for antigen B, and depending on what combination or lack of markers determines whether you have the other two in our blood typing. So again, these are glycolipids on our red blood cell surface. And again, the antibodies, which would bind to these two different types of antigens, are found in plasma of other blood types. So if someone is A blood type, then they'll make a marker against B. So only against what type of blood they're not is foreign to them. So we have an anti-A and an anti-B. So that's how we test the blood. If it clumps up in anti-A, then we know this is A blood type. If it clumps up in B, we know it's B. And we'll actually look at this, and you should have a Connect lab that does this. Okay, so let's look at our blood types. So we got 4 different types of blood that we focus on, our 4 major categories. Again, we have a ton of other types of markers, but they're just not as reactive as our ABO ones. So, blood type A, that person has A antigen, so A cell surface markers, also called agglutinogens. A B blood type has the B antigen, so B marker. AB has both. And type O doesn't have either one of them. So they lack these little extra tags on those glycolipids. So the most common blood type is type O without the marker. The rarest type is type AB. So having both markers on their red blood cell. So again, the antibodies, we can either make anti-A or anti-B depending on the type of it. It takes a second for those to start peering up, so maximum concentration not until about 10 years of age. But again, those antibodies are found in the plasma, and you do not form antibodies against your own antigen. So you're not a type A blood type making anti-A antigens. So not antigens, anti-A agglutinins or antibodies against a blood type, because then you'd be clumping up your own blood cells, and that's a bad thing. So you don't make antibodies against your own blood type. Okay, looking at our... Okay, cool. Just double checking on our study guide. Okay, so blood type, this is, you actually do need to know this chart. So what blood type and what antibody or agglutinins that they make. So if we have the agglutinogen A, it makes the B antibody against the B blood type. B makes anti-A antibodies, so the B blood type is going to have the B agglutinogen, but the A agglutinin. The O blood type doesn't have either the A or B agglutinogen, and so it's going to make both anti-A and anti-B agglutinin. So it's going to make antibodies against both of them. Type AB, which is our last major category, has both agglutinogen cell surface markers, but it's not going to make any antibodies against either one because it has both markers, and so therefore the blood would react if it did make a agglutinin against the A or B agglutinogen. So this should be on a later slide, but I'm going to say it now anyway because you might as well get it repeated. This makes blood type O because it doesn't have a marker that would interact with either of these two antibodies. So this is our universal donor. AB is the universal recipient because it doesn't make any of the antibodies, so you would have no response. The last type of antigen that we actually care about, because it is a possible reactive one, is the Rh factor, and it's just whether you're positive or negative for it. So there's different Rh factors. Some are different letters. The most reactive one is RhD, and that's the one we focus on. So either you have it or you don't. If you have it, of course, you're not going to make antibodies against it. But if you don't have it and you're exposed to Rh positive blood, so even if you're a AB blood type, but you're AB negative and you get AB positive blood, you will start producing that antibody. Now, that's not a big deal other than the fact of whatever blood is in your system is going to start clumping. But the bigger issue is that we focus on has to deal with pregnancy that we'll deal with later. So here is and again, you will do this on Connect. Post-COVID, we will do this in the lab to actually do this, except for it's with paint instead of real blood because, yes, some folks aren't that cleanly, and so we just do the paint version. But it still will have a reaction unless you have this blood type, and so I'll just let you do it again just so you can actually see the reaction. Okay, so we have two different antibodies. Again, we have our anti-A or anti-B. So we have 4 different samples. So on this one, it clumped in A, but not B. This one clumped in B, but not A. This one clumped in both. This one, no reaction at all. So this is how we're able to determine which of our blood types. So the one that clumped in A is type A. That's because we're testing against it. So because it clumped in that one and not the other one, we know that this is only type A, this is only type B. This one reacted with both, so it's both A and B. Again, the O marker has no cell surface agglutinogen, and so therefore it's not going to react to either of the agglutinins or antibodies. So that's how they determine your blood type if you, for example, donate. Okay, so let's say hypothetically you accidentally get the wrong blood. So the anti-A or B, whichever antibodies you have in your body, those are going to cause agglutination again, so that causes a clumping of the red blood cells. So here, the little green dudes, those aren't viruses, those are actually the antibodies. So the little sticks off the side of those help to bind to those cell surface markers, so those agglutinogens. So when they bind to multiple ones, and so all of this will start clumping up together as a blood clot. Now, this is a problem because our blood needs to flow freely, move around in all these ridiculously tiny little blood vessels. And this causes, it was called a mismatched transfusion reaction. So this goes into the question describing how they're active, how the agglutinogens and agglutinins are active in an immune response. So this is what we're going into. So our self-antibodies, so our agglutinogens will bind, so the ones that our body makes are anti every other blood type. Those, I just realized that, hold on, give me a Okay, let's try this again. So, mismatched transfusion reaction, you got the wrong blood. So, our self-antibodies, the one our body creates against the other blood types, so our own personal agglutinins, will bind to foreign agglutinogens. So this is the blood that has been donated and transfused into your body, so pumped into your own body. So the antibodies we make are going to bind to the red blood cells that are foreign, so causing our agglutination, so that clumping again. So because they are big clumps, and because we have these ridiculously small blood vessels, they end up blocking those blood vessels. And because they're trying to get shoved through these really tiny blood vessels, those foreign blood cells will hemolyze. So this is just a shorter version of lysis, so to break apart. So we're breaking apart those foreign red blood cells. Here's the problem. When that happens, the hemoglobin that's in those red blood cells will be released from those red blood cells and into the bloodstream. So as it circulates, again, some of your blood vessels, it's fine, but then that big chunky protein of a hemoglobin gets to really small structures. So we'll talk about, again, exam 9 section, dealing with the kidneys and urine production. So those hemoglobin, those big chunky molecules, as they get into the really tiny structures of your kidneys, it could block those and causing renal failure. So basically your kidneys fail, you can't make urine, you can't get that waste out, you can't get that excess fluid out, and you're going to have a major problem. So you would probably start having to get dialysis until you get dealing with your situation. So anyway, causing your kidneys to fail. Oops, go back. Okay, so dealing with our different blood types. So our universal donor, that is type O, because they do not have those cell surface markers. They do not have those antigens antigens that would identify them as a foreign blood cell. So they're able to kind of sneak in and help the person have enough blood cells. The plasma in the donor, so the blood that you're receiving, may have antibodies against the recipient's red blood cells. So what they do is, a lot of times when you give blood, they'll filter out the plasma and just focus on the red blood cells, which is what you need. And so therefore, minimizing the plasma, minimizing the antibodies that would be in the donor's blood, and so therefore minimizing the possibility of a reaction. Okay, universal donor, that's type O because it's not going to have a foreign flag on it. The universal recipient is type AB, again, because it doesn't make any of the antibodies, it doesn't make any of those agglutinins, and so therefore no reaction. So the last thing is our RH factor, so whether or not something is positive or negative. So there's a couple of different ones. Our RHD factor is our most reactive, and that's the one that we focus on, but only in dealing with situations of pregnancy. So if you have that RH factor, that's just an extra foreign flag on a red blood cell, and so that can actually be detected. So normally you don't have antibodies or agglutinins against that RH factor, but only if you are exposed to RH positive blood. So it could be O positive, A positive, B positive, AB positive. If you are positive for that RH factor and that, if you're negative for the RH factor and you get exposed to that type of blood, so that's why all the times, you watch the hospital TV drama shows and they're like, we need O negative blood, universal donor blood without this RH factor. Okay, so if you're exposed to that blood, that's when you start making antibodies against that RH factor. So either through a transfusion, usually the situation happens when there's a pregnancy. So the mother is RH negative, and let's say her fetus has RH positive blood. So no problems the first time. It comes the second time with a possible exposure. So up top, here we have the mother who has now been exposed to a RH positive baby. So her body creates antibodies or agglutinins against the RH factor. So the next time she gets pregnant with a, of course, same blood type baby, then the antibody she created here will now attack the baby. That's not good. So in order to prevent the production of these antibodies, if it is determined that the fetus is positive, but the mother is negative, then a shot called Rogam is given, and that prevents the production of the antibodies. So no anti-D antibodies are created, so therefore the baby is safe on the second pregnancy. So I heard this shot is very painful, but hey, at least it allows you to have multiple children, because the person I have it, she's actually, I think they're at 3 kids. But she's doing good. She finished LPN school and now about to start RN school. So, things are looking up. Okay, great. Okay, so still able to do it with 3 very energetic children, but they're adorable. Okay, so this is the end of this section. So we'll start another video. Yes, I know, unfortunately, this is long, but hey, you got to learn all this stuff. See you next time.